Hello, BISC1323. This is the beginning of recorded lecture 1.5. We're continuing on with these 6 eukaryotic supergroups, and we're going to finish it up in this lecture. So we did 3 of them in the previous lecture. We're going to finish the other 3 now. So moving on, our next one is this group called Rhizaria. So there's really nothing easily identifiable about Rhizaria as a supergroup, so we're just going to talk about the 3 groups within it. First up, we have forams. Forams are amoeba with thin pseudopodia. Look familiar? Sound familiar? Our last group in the previous recorded lecture, nucleariids, were also amoeba with thin pseudopodia. This is an example of convergent evolution. We're going to see this a lot, as I keep saying. Different supergroups of eukarya, they just happen to have converged upon a similar body plan. So yeah, forams are rhizarians. They're amoeba with thin pseudopodia. There is something interesting about these forams, though. So they form tests, and that's also not something new. We saw tests in an earlier group as well. That's more convergent evolution. But part of the key term's definition of test is that these shells were made of organic compounds. They're carbon-based. So when these forams die, their tests sink to the bottom of the ocean and act as carbon sinks. So yeah, here's some of the diversity of these tests. So this is trapping up carbon in these tests, and then it eventually sinks down, becomes mineralized into rock. This is a way of taking carbon out of an ecosystem, putting it into solid form, putting it into rock. The term we use for this is a carbon sink. So that's the important thing about these forams and their tests. Another rhizaria is radiolarians. These are also amoeba with thin pseudopodia. Here's a shell by itself. You can kind of see the holes in it where those pseudopodia would have come out of, but otherwise it kind of looks like all the other amoeba with thin pseudopodia. Now, interestingly, I call this a radiolarian shell, not a test. A test is specifically a structure made of organic compounds, as I just explained, the whole carbon sink thing. What we call a shell is not made of organic compounds. These shells that radiolarians make, they fulfill a similar function as the tests, but they're made of silica. It's basically sand, basically glass. And these preserve very well as microfossils. So I'm not this kind of paleontologist, but apparently looking at microscopic fossils, looking at the different shapes and sizes and popularity of different types of radiolarians. Again, this is outside of my area of expertise, but the right type of paleontologist can identify what period of time that fossil was from based on what sort of shapes that we see in the radiolarians in the fossil. Cool stuff, but all I'm going to say is that they're useful indicators of environmental conditions as well as fossil record time periods. So yeah, they change with the environment, so it's a cool way of having a window into the past, of looking at these preserved microfossils of these silica shells. And the third group of rhizarians is sarcozoans. This is a true grab bag, and some of these sarcozoans have a test, some of them don't have a test. Some of them have a shell, some of them don't have a shell, and hopefully you appreciate the difference between a test and a shell. They have all different shapes and sizes. It's kind of hard to nail down what they do. We really only know that this is a group and that members of this, with tests and shells are not instead radiolarians or forams. We only know that it's a distinct group here because of genome sequencing and sequence alignment, sequence comparison, all that stuff I talked about in the first lecture. So what can we say about sarcozoans? Diverse forms, and some of them are actually photosynthetic. So this is the first photosynthetic protist that we've seen. This means there was an endosymbiosis here in these cercozoans for some of them to gain this ability to do photosynthesis. Now, wait a second. This isn't the first time that we've seen endosymbiosis. We saw this in Archaea plastida. We saw this in plants and in red algae in this shared group. So what gives? How are we seeing photosynthesis twice? Well, spoilers, we're going to see it a couple more times when we get into the other supergroups. This endosymbiosis of a photosynthetic bacterium gaining a photosynthetic plastid is not something that just happened once, like with mitochondria. It happened several times in several different lineages and several different supergroups of eukaryotes. This is another example of convergent evolution, separate groups of eukaryotes doing the same thing completely independently of one another. So, yep, some of these cercozoans are photosynthetic, separate endosymbiosis event from the endosymbiosis that gives photosynthetic capabilities to plants or the other ones that we'll talk about later. So that's interesting, I think. Photosynthesis convergently evolved multiple times in eukarya. That's our take-home message. Not all of them are photosynthetic, though. Some of these cercozoans, to continue to add to their grab bag of features, some of them are actually parasitic. Nothing we need to worry about, but they're parasites of fungi. I think this is interesting because we've seen thin pseudopodia a couple of times already. And when I first described these structures, I said they increase surface area so that you can absorb more nutrients from the water. Well, in this group of cercozoans, I like this genus Vampirella, they actually use these thin pseudopodia to stab into their fungal hosts and then steal nutrients from within the fungi's cytoplasm. So an offensive use of these thin pseudopodia. I think it's worth noting for these cercozoans. They steal nutrients from the cytoplasm of their host using needle-like pseudopods. Okay. That was Rhizaria, cercozoans, forams, and radiolarians. We got two more groups, but this next one's a big one. This chrome alveolata. In fact, this is so big, we could see that within this supergroup, there are two smaller groups, alveolates and straminopiles. By the way, as a bit of an aside, I may have a test question or two asking, which of the following is not a part of this supergroup, or here's forams, which supergroup is it a part of? But honestly, I think what I really want to focus on, as far as your understanding, again, a little bit of which are members of which supergroups, but most of what I want you to focus on when you're studying is, for each of these individual things, what's their deal? What's the deal with choanoflagellates? What do they do? What are their unique features? What's the deal with cercozoans? What do they do? What are their unique features that I've described in these slides? So don't get too intimidated. Again, I may have a question or two about what's an alveolate or a straminopile or things like that, but I would focus primarily on what's the deal of each of these individual groups. So, okay, chrome alveolata. What are we doing here? Chrome alveolates are photosynthetic. Again, this is the third time we've seen this, although we'll see examples. Some have lost this ability. So this is another thing that happens interestingly in evolution. We're going to see some of these that are photosynthetic, some of these that are not. As a lineage, their ancestor was photosynthetic, but just like some birds have lost the ability to fly, and some reptiles have lost their limbs to become snakes, some of these chrome alveolates have lost their photosynthetic capabilities. So, as I said, there are two groups within chrome alveolata because this is so big. There's the alveolata and the straminopiles. Let's take on alveolate. So, chrome alveolata, within that, alveolates, within that, dinoflagellates. So what can we say about dinoflagellates? Well, dinoflagellates are mostly marine. I believe this is the first time this word has come up in conversation. Key terms definition of marine is an organism that lives in the high salt water of an ocean or sea. So marine just means lives in the ocean. And dinoflagellates are plankton, which is also an important key term. So I think most people hear plankton, they think of something like this. Plankton is not a species. It's not a specific type of thing. It's a broad category of different things. If something is described as plankton, it just means it lives in the water and it goes with the flow. It's not actively swimming or anchored to a structure. It's just going along with the Wasserströmung. Sorry, the best figure for this was in German. This just means water current. It means going with the flow. The key terms defines plankton more concisely. Plankton is a diverse group of mostly microscopic organisms that drift in marine and freshwater systems. So we're going to see plankton again in other groups, but yeah, it means floating and drifting. So what else can we say about dinoflagellates? Well, there are several important notable members of dinoflagellates. Some of these produce toxins in the water responsible for red tide, very dangerous algal bloom. This one's in Florida. Stay away from this. So these are dinoflagellates doing this. Some dinoflagellates are bioluminescent. Despite looking pretty crazy, this is not harmful to you. It just looks pretty, producing this fluorescent light. So yeah, some make toxins, some are bioluminescent. Another very important group of dinoflagellates are part of corals. We'll talk way more about corals when we get to animals because these things are animals, but they kind of live like plants because they have photosynthetic partners that they wholly rely upon for their survival, for their nutrients. So these photosynthetic partners inside of the animal coral are protists and they're dinoflagellates. So some are photosynthetic partners of corals, which are animals. So yeah, all three of these things are different examples of what dinoflagellates can do. This chrome alveolata alveolates also includes a group called apicomplexans. And yeah, you know you're in for a wild ride when the name of the group has complex as part of its name. So what do I mean by complex? I mean something like this. So we are not going through this step by step, but it is interesting to look at. This is the life cycle of the protist parasite responsible for malaria. And yeah, you can see there are several stages inside the mosquito, different types of reproduction, several stages inside the human, some specifically in the liver, some that only happen in the blood, other types of sexual and asexual reproduction and stuff like that. Yeah, complex life cycles. And it's not just malaria, there are others here as well. I'm just going to say all of these apicomplexans are parasites with complex life cycles. Because malaria has such a big impact on public health, I do want to call out its species by name. Apicomplexans includes Plasmodium spp, which causes malaria. I think this is the first time we've seen this. So I introduced the binomial nomenclature in the first chapter, genus and species. Plasmodium is the genus. SPP is not a particular species. When we see this abbreviation, it means several species within this group. So there's Plasmodium falciparum is one of the big ones, but there are several different species within Plasmodium that cause malaria. So when we want to refer to several species, we use SPP. Anyway, it's an apicomplexan. Also within Chromialveolata alveolates, we have a group called ciliates. And again, the names themselves do a pretty good job of describing what they are. Ciliates, as you can imagine, are covered in cilia. They use cilia to swim around, and yeah, they're gobbling up bacteria, and yeah, not much else to say about these ciliates. Covered in cilia, and yeah, they're heterotrophs. They eat bacteria. So these were the alveolates within Chromalveolata: dinoflagellates, apicomplexans, ciliates. Now, we have straminopiles, and there are going to be 4 straminopiles, which are, again, we're still part of Chrome alveolata. So what's the deal with straminopiles? They have hairy flagella. What's a hairy flagellum? Oh, okay, yeah, I guess that checks out. So yeah, many of them have this distinct flagellum. Again, plenty of other groups have flagella. Flagella is not unique to straminopiles, but many of them have this distinctive hairy flagellum. So what about the groups within this? So within straminopiles, first up we've got diatoms. Diatoms are very pretty. So once again, we're talking about a group of protists that makes this protective shell-like structure. This is not a test. These shells are made of silica. We've seen that before, so basically, glass or sand. And yeah, they come in lots of different diverse shapes and sizes. Yeah, diatoms, they're photosynthetic, unique complex shells of silica. And this is another thing that is plankton, because these diatoms are not actively swimming around. I know I just said they have hairy flagella. Not everything in straminopiles has this hairy flagellum. It's just something to try to identify them. But these diatoms float around in the water, and they're doing photosynthesis. Another straminopile is a group called golden algae. Oh, okay, that's kind of golden looking. So these do photosynthesis. They are plankton, and they can form these, kind of looks like a bundle of wheat, I guess. They can form these colonies. And once again, this is not a multicellular organism. This is a colony of individuals. We've seen that several times in protists. So golden algae, photosynthetic plankton, but unlike diatoms, which roll alone, they can form these branching colonies. Also, within straminopiles, we have brown algae. Now, just kind of an aside, algae is one of those terms that doesn't really mean much. We've got brown algae, we just had golden algae. If you go back to the beginning of this chapter, we had red algae, and when we get to plants, there's going to be green algae. Algae, and there's even a group of bacteria that is blue-green algae is sometimes used to describe it. So the word algae by itself kind of just broadly refers to something that lives in the water and does photosynthesis. And yep, certainly red algae lived in the water and did photosynthesis. Golden algae lives in the water as plankton, does photosynthesis. And brown algae lives in the water, does photosynthesis, but a little bit differently than the golden algae. Brown algae is big. Brown algae is macroscopic. You can see this with the naked eye. In fact, if you've visited almost any beach in the world, you've probably seen stuff like this washed up on the beach. That's brown algae. A lot of people would see this on the sand and say, oh, this is some sort of plant. I mean, these obviously look like leaves. These kind of look like roots. It does photosynthesis. This must be a plant, right? Well, we know that it's not. This is a protist. The reason why this looks like a plant is convergent evolution. So this group of protists and plants came up with the same kind of leaf-like structure and stem-like structures because it works. If you're doing photosynthesis and you're going to be a big macroscopic organism, having these leaf-like structures are a great way to absorb as much light energy as possible. So it is convergent. They convergently evolved to look like a plant, but these are not plants, they're protists. They're also known as seaweed, also known as kelp, multicellular. We haven't seen multicellular in a while, but yeah, these are multicellular and photosynthetic, and the phrase I'm using is that they are superficially similar to plants. They look like plants, but they're not. All right, one more group of straminopiles, and that is oomycetes. So oomycetes are saprobes or parasites. New word alert. So what is a saprobe? Well, a saprobe is this. The textbook assures me that there's an insect underneath all of this horrible white stuff. A dead insect, I should say. Saprobes are, let me just read the key terms definition. A saprobe is an organism that derives nutrients from decaying organic matter. So yeah, dead bug growing on top of it, taking nutrients from its body. That sounds a lot like fungus. I know we haven't gotten to the fungus chapter yet, but this whole saprobe thing of breaking down decaying organic matter, that's a fungus, and this looks a lot like some sort of horrible fungal mold. These oomycetes are definitely superficially similar to fungus. They look like a fungus. They have the lifestyle of a fungus. But obviously they're not fungus. They're part of this group of protists, these chrome alveolates. This is yet another example of convergent evolution. Two completely, very distantly separate groups coming up with the same idea, the same lifestyle, independently of one another. And in fact, these were classified as fungi for quite a long time until modern genome sequencing technology let us know, hey, weirdly enough, these things breaking down dead organic matter, they're more closely related to kelp than they are to any fungus. So weird discovery, but hey, that's how evolution works. There is a notable member of this oomycetes, and that is Phytophthora infestans. Again, a very evocative species name, infestans. This is responsible for potato blight. So yes, this is the Irish potato famine, huge, global political and economic repercussions, not just on Ireland, but across the world. This potato famine was caused by this potato blight. Again, not a fungus, destroying these crops, but this group of protists responsible for potato blight. All right, we are closing in on the end of this list of supergroups here. We just finished chrome alveolata, really big one. There's just one supergroup left up here in yellow, the excavata. So what can we say about excavata? Well, these are all single-celled, so no more big stuff like the kelp. And sometimes the names are evocative. It's a way to kind of remember what they look like. But apparently excavata, this group was named for a feeding groove that they have in the side of the cell that someone thought looked like it had been excavated from the side of the cell, hence excavata. I don't get to name these things, but I try to draw these connections. Here's a, not very detailed drawing of one of these. There's a nucleus, and yeah, the excavated feeding groove is this this structure right here. We'll see it again in another couple of slides, but yeah, that's where excavata comes from. So there are three groups within this. Let's start off with diplomonads. So once again, the name is kind of a clue to what their deal is. It's got di right here at the name. Di as a prefix means two. So these things are going to have two of something. What are they going to have two of? No, these are not eyeballs. Remember, these are single-celled organisms. They're nuclei. These diplomonads have two nuclei in each cell. And these nuclei are not two copies of the same thing. They're non-identical nuclei. The reason why they keep their chromosomes in two separate nuclei instead of in one, like everyone else does, is not well understood. So we don't know why they're doing this. There's another weird feature to their cells that you can't see from the microscope image, but if you look deeper at what they've got going on the inside, they don't have mitochondria, which is weird. That was one of the things at the very beginning of the chapter that we said, all eukaryotes have. They seem to have lost their mitochondria, or at least reduced them in size to structures called mitosomes. And just like the weird double nuclei, the reason for this is not well understood. So diplomonads have two non-identical haploid nuclei. They have reduced mitochondria that we call mitosomes. We don't know why they're doing either of these things. And there is one important notable member here, Giardia lamblia. This is what we were looking at here, incidentally. Giardia lamblia is responsible for giardiasis, also known as hiker's diarrhea. So Not the most life-threatening of things, but yeah, the next time you're off hiking somewhere and you see this cool, crisp, cold mountain stream, there's a good chance a deer pooped in that half a mile upstream, and there's giardia swimming in it. So yeah, be careful, be safe when you're hiking, because yeah, this protist causes hiker's diarrhea. Okay, what else do we have in excavata? Well, next up we've got a group called parabasalids. So parabasalids, here's one. Yep. Single nucleus, okay, so we're back in a world that makes sense with this. Single nucleus. We see some flagella here for swimming around. Here's that feeding groove that excavata is named for. And this looks like another flagellum here at the other end of the cell, but this it does not flap around like flagella. This is called an axostyle, and it's used for stabbing. It's used for attachment. And usually when you're talking about some microorganism that has a structure for attachment, it's usually up to no good. It's usually a parasite or a pathogen if it's trying to attach to a host. And indeed, that's what's going on with this one. So parabasalids also have reduced mitochondria, just like we saw for the diplomonads. But they also have flagella and this axostyle for attachment. Again, attachment usually means they're up to no good. The one we were looking at here is a notable member, Trichomonas vaginalis. Trichomonas vaginalis, again, genus, species, causes trichomoniasis, an interestingly common sexually transmitted infection. I'm just going to rattle off some stats that you don't need to memorize, but about 70% of affected people with trichomoniasis don't have any symptoms at all, which allows it to spread effectively because you're not getting treated or cured if you don't have any symptoms. It can lead to complications during pregnancy and increased risk of contracting HIV. Despite vaginalis as the species name, a vagina is not required to get this. Anyone can be infected with Trichomonas vaginalis. It's apparently the most common non-viral sexually transmitted infection in the United States, with 3.7 million prevalent cases per year. And so just some interesting stats, but no, sexually transmitted infection, Trichomonas vaginalis. Now, the axostyle usually means you're up to no good, but there's actually one other example of a parabasalid that is not using this axostyle for evil. It's Trichonympha SPP, and there's the SPP again. These Trichonympha are attaching to the walls of the termite gut and helping these termites break down cellulose. So they're associated with the termite gut, but they're not parasites. They're living in a mutualistic symbiotic relationship where they break down cellulose that these termites consume. They take some nutrients to themselves and give a lot to these termites. So they're also, I think, a very notable member here, Trichonympha, living in the termite gut, breaking down cellulose. Okay. One more group of excavata here, euglenozoans. Euglenozoans have flagella, and they are freshwater mixotrophs. Finally, here at the end, I told you we would see a mixotroph. So yeah, here's a typical euglenozoan swimming around in the water using its flagellum, bringing bacteria that it eats into this reservoir, which is basically a mouth where it can, eat the stuff and absorb nutrients. But in addition to that, chloroplast, it's doing photosynthesis. So exactly what I described a mixotroph would be doing. These are probably the most typical ones, swimming around in the fresh water, doing that stuff, but there are also parasites within this group of euglenozoans, notably one called Trypanosoma brucei, which is responsible for African sleeping sickness. So here's Trypanosoma brucei, not much to look at, lean, mean, single-celled organism, and when I was talking about protists in general, it's not just apicomplexans that have complex life cycles. A lot of these parasites have complex life cycles, and here, don't sweat all the details here, but yeah, this is Trypanosoma brucei. African sleeping sickness is the disease that it causes. So, okay, we've finished with all 6 of these eukaryotic supergroups at this point. There is one last thing that I want to say in this chapter before we can finally be done with protists, and that is the following statement. It has to do with the word protozoa. I haven't used this word. I don't like using this word, but it's something you may come across in other classes or other places. The word protozoa is used to refer to single-celled heterotrophic protists. That is a lot of stuff. Like, here's a grab bag of things that we've seen in this chapter. All of these things are single-celled. All of these things are heterotrophs, and they're all protists. So these can broadly be referred to as protozoans. If we want to identify what the protozoans are, it looks like this, and oh my goodness, this is like almost every supergroup here is included. Almost every supergroup. They're across the board here. This is definitely not a clade. This is definitely not an evolutionarily significant group. It's another sort of grab bag term, but I mean, it kind of makes sense. They look like they all must be closely related because they have a very similar lifestyle, so that's where the name came from. But again, we're all trying to phrase out the use of protozoa because it's not evolutionarily significant at all. It's not a clade, but it's something that you may run into. So I want you to at least be familiar with what it means. Okay, with that, we come to the end of the protist chapter, the end of lecture 1.5, and this is the cutoff point for exam number 1.